Welcome back to the deep dive.
Good to be here.
So today we're tackling a really intricate system. We're using Porth's Pathophysiology, Chapter 42, as our guide.
That's right.
We're looking at the male genitourinary system, its structure, how it works, and what happens when things go wrong.
Yeah, it's a fascinating setup. You've got genetic triggers, really precise temperature needs, a whole hormonal feedback system.
A lot to unpack.
It is. But our goal really is to map it out for you. We want to look at the architecture, trace those hormonal pathways.
And critically, connect it all back to clinical conditions, right?
Yeah.
Like hypogonadism, BPA.
Exactly. Connecting the underlying mechanism to what you actually see in a patient. That's the key.
Okay.
And just to lay the groundwork, functionally, you can sort of think of four main jobs.
Right.
The tests are for making hormones and sperm. Then you have the ducts for transport and storage.
Mm-hmm.
Accessory organs make the seminal fluid. And the penis handles elimination and sexual function.
Okay, let's start at the absolute beginning then. How does this whole system even get built? The origin story, like you said.
Yeah, it's pretty amazing. In the embryo, early on, you actually can't tell the difference.
Doing male and female tracks?
Nope, they're indistinguishable. It's not until around the 7th week of gestation that things start to differentiate.
7 weeks. Okay, so what's the trigger?
It all comes down to genetics, specifically the SRY gene.
SRY.
Yep, located on the Y chromosome. If it's present and active, it basically flips the switch.
Telling the gonads to become testes.
Exactly. Without that SRY signal, the default path is for the gonads to develop into ovaries.
So before that switch, the embryo has potential for both.
It does. It has two sets of ducts initially, the Wolffian ducts.
Those are the male precursors.
Right, and the Mullerian ducts, which are the precursors for female structures like the uterus and fallopian tubes.
So for male development, you need to encourage one and get rid of the other.
Precisely. It's a two-pronged hormonal strategy launched by the newly formed fetal testes.
Okay, hormones. What are they?
First, there's anti-Mullerian hormone, AMH. Its job is pretty clear from the name. Suppress the Mullerian ducts.
Stop the female structures from forming.
Correct. At the same time, the testes start producing testosterone, or T.
And that stimulates the Wolffian ducts.
Yes, it tells them, okay, develop, and they become the epididymis, the vas deferens, the seminal vesicles, the internal male plumbing.
But wait, what about the external parts? And the prostate?
Ah, good point.
Right.
That requires another step. Testosterone gets converted locally into a more potent androgen called dihydrotestosterone, DHT.
DHT, okay.
This conversion happens thanks to an enzyme called 5-reductase, and DHT is the key player for developing the prostate and all the external male genitalia, the penis and scrotum.
So you absolutely need both T and DHT for full masculinization.
You absolutely do. And Porth makes this really clear. Let's say you have an XY embryo, so it has the SRY gene, the testes form, they make AMH.
So the Mullerian ducts regress.
Right. But what if there's a problem making T? Or maybe the 5-reductase enzyme isn't working, so you can't make DHT.
Yeah.
Then the Wolffian ducts don't get the signal to develop, and the external structures don't masculinize either.
So you end up with what?
You end up with female external genitalia. Because without that active androgen signal, development just defaults to the female pattern externally.
Wow. So it's not automatic at all. It requires this active hormonal push.
Exactly. Development defaults towards female unless actively driven otherwise by these specific androgens at the right time.
Okay, moving on from development, let's talk about the tests themselves. Their location is unique, outside the body.
Yeah, in the scrotum. And there's a very, very critical reason for that. Temperature.
Right. I remember reading this. Sperm production doesn't like normal body heat.
Not at all. It works best optimally at about 2 to 3 degrees Celsius cooler than core body temperature.
So how does the body manage that? Keep them cool enough?
It's got a couple of clever mechanisms. First, muscles. You've got the dardos muscle in the scrotal wall and the cremaster muscles attached to the testes.
And they react to temperature.
They do. When it's cold, they contract, pulling the testes up closer to the body to warm them up.
And relax when it's warm, letting them hang lower.
Exactly. It's like a little elevator system for temperature control.
Okay, that's one part. What else?
The other part is this amazing vascular network called the pampiniform plexus.
Pampiniform plexus.
Yeah, it's basically a mesh of veins wrapped around the testicular artery as it heads down to the testes.
Like a radiator.
Sort of. It works as a countercurrent heat exchanger. The cooler venous blood returning from the testes absorbs heat from the warmer arterial blood coming in.
So the blood reaching the testes is already pretty cool.
Precisely. It's incredibly efficient.
And if this system fails, like if the testes don't descend properly, That's cryptorchidism, right?
Correct. Cryptorchidism, or even things like wearing very tight underwear consistently, or prolonged fevers.
Yeah.
Anything that keeps the testes too warm can impair that cooling.
Leading to problems with sperm production.
Yes, potentially decreased sperm counts and even infertility. It highlights how vital that temperature regulation really is.
Okay, so assuming the temperature's right, where do the sperm go after they're made? Let's trace the path.
All right. Production happens deep inside the testes, in the seminiferous tubules.
Okay.
From there, they move into the epididymis. And this is a crucial step.
Why's that?
Because when sperm leave the tubules, they can't really swim or fertilize yet. They mature and gain motility during their journey through the epididymis. It takes time.
So epididymis is like sperm boot camp.
Huh, you could say that, yeah.
Hmm.
Once they graduate, they move into the ductus deferens or vas deferens.
Which is basically a transport tube.
A muscular one, yeah. It propels them along. And the final section of the vas deferens, near the prostate, widens into the ampulla.
Ampulla. And that's for storage.
That's the main storage depot for mature, ready-to-go sperm.
Ah, and that connects to vasectomy, doesn't it?
It does directly. When a vasectomy is done, the vas deferens is cut.
But the sperm already passed the cut in the ampulla.
They're still there. That's why a man remains fertile for usually about 4 to 5 weeks after the procedure. The stored sperm needs to be cleared out.
That is such a critical piece of information for counseling patients.
Absolutely essential.
Okay, so sperm are produced, matured, stored. What about the fluid they travel in? Semen. That comes from the accessory organs, right?
Correct. Semen is sperm plus secretions from 3 main glands: the seminal vesicles, the prostate, and the bulbourethral glands.
Let's start with the seminal vesicles. They make most of the volume.
They do, about 70% of the ejaculate volume. And their fluid is really important. It contains fructose.
Sugar for energy.
Exactly. Fuel for the sperm's long journey. It also contains prostaglandins.
And those affect the female tract.
It's not so, yeah. They might help by altering cervical mucus or maybe promoting slight contractions to help sperm travel upwards.
Okay. What about the bulbourethral glands? They're also called Cowper's glands.
Yep, Cowper's glands. They're small, located near the base of the penis. They secrete a clear alkaline mucus.
Before ejaculation.
Right, usually during arousal. The idea is it lubricates the urethra and, importantly, neutralizes any acidic urine residue that might be left. Cleans the pipes, basically.
Preparing the way. And then the prostate.
The prostate. It sits right below the bladder, wrapping around the urethra.
And its secretion.
Is a thin, milky, and very alkaline fluid. It contains things like citric acid, some enzymes, but the key thing is its alkalinity.
Why is that so crucial?
Because the fluid coming from the vas deferens is actually slightly acidic, and the vaginal environment is also quite acidic.
So the prostate fluid acts like a buffer.
Exactly. It neutralizes both of those acidic environments, bringing the final pH of the semen into that narrow optimal range around 6.0 to 6.5.
And that pH is needed for the sperm to become fully mobile.
Absolutely critical for sperm motility and survival. Without that prostatic fluid buffer, they'd be immobilized or damaged by the acidity.
But that location, wrapping around the urethra, that's where problems arise, like BPH.
Precisely. Benign prostatic hyperplasia, or BPH, is non-cancerous enlargement of the prostate gland. It's very common in older men.
And because it surrounds the urethra.
When it enlarges, it squeezes the urethra. It physically obstructs the flow of urine out of the bladder.
Like the example on Porth, Mr. Topers.
Exactly. His symptoms are classic BPH. Trouble starting urination, a weak stream, dribbling, feeling like the bladder isn't empty, needing to go frequently.
Because the outlet is partially blocked.
Just like stepping on a garden hose, the bladder muscle has to strain against that resistance.
Makes sense. And just quickly, the penis structure itself.
Right. It's composed of 3 columns of erectile tissue, 2 corpora cavernosa side by side.
Those are the main erectile bodies.
Yes, they do most of the engorging with blood to create rigidity.
Right.
And then there's 1 corpus spongiosum underneath.
And the urethra runs through that one.
Correct. The corpus spongiosum surrounds the urethra. It also engorges, but less rigidly, which is important to keep the urethra open during erection so ejaculate can pass through.
Okay, structure covered. Let's shift to the control system. The hormones. This all kicks in at puberty, right?
Right.
Around 10 or 11.
Generally, yes. That's when the main hormonal control center, the hypothalamic pituitary gonadal axis, or HPG axis, really wakes up.
The HPG axis. It's like a classic endocrine feedback loop.
It is a really elegant one. You can think of it starting at the top, in the brain.
Hypothalamus.
Right. It starts releasing gonadotropin-releasing hormone, GnRH.
Impulses.
Yes, the pulsatile release is important. GnRH travels just a short distance down to the anterior pituitary gland.
And tells the pituitary to release.
The gonadotropins, luteinizing hormone, LH, and follicle-stimulating hormone, FSH.
LH and FSH. Okay, pituitary level done. Where do they go?
They travel through the bloodstream down to the testes, where they each have specific target cells.
Different jobs for each.
Different jobs. LH primarily targets the Leydig cells, which are found in the spaces between the seminiferous tubules.
The LH tells the Leydig cells to do what?
Produce testosterone. LH is the main driver of testosterone production.
Okay. And FSH?
FSH targets the Sertoli cells, which are inside the seminiferous tubules, kind of nursing the developing sperm cells.
So FSH is involved in sperm production.
Correctly. It initiates spermatogenesis. The Sertoli cells, stimulated by FSH, also produce another hormone called inhibin.
Inhibin. Okay, so we have GnRH, LH-FSH, testosterone, and inhibin. How does the feedback work?
It's negative feedback. When testosterone levels in the blood get high enough.
They signal back up the chain.
Exactly. High T tells the hypothalamus to slow down GnRH release and tells the pituitary to slow down LH release.
So it's the brakes on its own production.
Right. And inhibin, produced by the Sertoli cells in response to FSH.
So it's the same for FSH.
Specifically for FSH. Inhibin feeds back to the pituitary to selectively suppress FSH release without affecting LH much. It fine-tunes the sperm production side.
A very neat, self-regulating system.
Keeps everything within the right range.
And testosterone itself. It does a lot more than just sperm production, obviously.
Oh, absolutely. Porth's chart 42.1 lists its functions. We already mentioned fetal differentiation.
Right, driving male development.
Then there's maintaining the primary sex characteristics, libido, and triggering the secondary characteristics at puberty, deepening voice, hair growth patterns, etc.
And the anabolic effects you mentioned. Building muscle.
Huge anabolic effects. It promotes protein synthesis, builds muscle mass, increases bone density. That's why boys gain, on average, about 50% more muscle mass during puberty compared to girls.
Okay, so if this whole axis or the tests themselves aren't working right, that leads to hypogonadism.
Correct. Hypogonadism just means reduced function of the gonads, either low testosterone production, low sperm production, or both.
And classifying it is key for figuring out why it's happening.
Absolutely. The key is looking at the hormone levels together, T, LH, and FSH.
So primary hypogonadism.
Primary means the problem is in the tests themselves. They aren't responding properly.
So testosterone and a sperm count would be low.
Right, but the pituitary is working fine. It senses the low T, so it ramps up production of LH and FSH, trying to stimulate the failing testes.
Ah, so the signature is low T but high LH and FSH.
Exactly. Causes could be things like damage from mumps orchitis or genetic conditions like Klinefelter syndrome, where you have an extra X chromosome, 47,000 XXY.
Okay. What about secondary or tertiary?
Secondary means the problem is in the pituitary. Tertiary means the problem is in the hypothalamus. Often they're grouped together because the result is similar.
Which is?
The pituitary isn't sending out enough LH and FSH signals in the first place, so the testes, even if healthy, don't get the command to produce.
So you get low T and low sperm.
But this time, because the stimulating hormones are missing, LH and FSH levels are low or inappropriately normal. They aren't elevated like in primary.
That distinction seems really important diagnostically.
It's crucial. It tells you where to look for the cause.
And clinically, what does adult-onset hypogonadism look like?
The symptoms can be kind of vague sometimes, which makes diagnosis tricky. Fatigue, low energy depression or low mood, decreased libido, maybe loss of muscle mass, increased body fat, sometimes erectile dysfunction.
Right.
Yeah.
And the source mentioned something really important here, the link to cardiovascular health.
Yes. This is a major point. There's growing evidence that low testosterone and also erectile dysfunction can be early warning signs.
For heart disease.
For cardiovascular disease, yes, along with things like type 2 diabetes. These conditions often travel together.
So if a patient presents with symptoms of hypogonadism, or especially ED,
a thorough cardiovascular workup is essential. It's not just about the reproductive system anymore. Low T can be a marker for underlying systemic issues like inflammation, insulin resistance, poor lipid profiles, all contributing to heart disease risk.
That's a really powerful connection. Okay, let's shift gears slightly to the actual mechanics of the sexual act. Neural control.
Right. This involves a neat interplay between the two branches of the autonomic nervous system.
Parasympathetic and sympathetic. Which one does erection?
Erection is parasympathetic. Think point P for parasympathetic, P for point.
Okay. Parasympathetic for erection. How does it work?
Stimulation travels via the pelvic nerve. The key chemical messenger released at the nerve endings in the penis isn't acetylcholine or norepinephrine, like usual for autonomics.
It's something different.
It's nitric oxide, NO, that same molecule involved in blood vessel dilation elsewhere.
Ah, so NO is released.
And it causes the smooth muscle in the walls of the small arteries and within the erectile tissue, the corpora cavernosa, to relax.
Relaxation lets blood flow in.
Rapidly. It shunts blood into those cavernous spaces. They engorge, compress the veins that drain blood out, and that trapping of blood causes the erection and rigidity.
Okay, parasympathetic and NO for erection. What about emission and ejaculation?
Orgasm. That's the sympathetic nervous system's job. Think, shoot. S for sympathetic, S for shoot.
Point and shoot. Got it.
Right. Sympathetic nerves, mainly from the L1 and L2 spinal levels, take over. First is emission.
What's emission?
That's the process where the sperm from the ampulla plus fluids from the seminal vesicles and prostate are moved forward into the urethra at the base of the penis. Critically, the sympathetic signals also cause the internal urethral sphincter at the bladder neck to close tightly.
To prevent.
Retrograde ejaculation signal going backwards into the bladder instead of forwards out the urethra.
Okay, so emission loads the chamber, then ejaculation.
Ejaculation is the forceful expulsion of that semen from the urethra. It's caused by rhythmic contractions of the accessory glands themselves and also the bulbospongiosus muscle at the base of the penis, plus other pelvic and trunk muscles. It's a sympathetic reflex.
A very coordinated handoff from parasympathetic relaxation to sympathetic propulsion.
Exactly.
All right, finally, let's touch on aging. Things change over time, obviously, sometimes called andropause.
Yeah, although it's not as abrupt as menopause in women. It's a much more gradual decline involving the endocrine system, circulation, even neuromuscular changes.
And testosterone levels do decline.
They do, gradually. Porth mentions an estimate of maybe a 10% drop per decade after around age 25 or 30. Yeah. It varies a lot between individuals, though.
What are the physical changes associated with that?
Well, the testes tend to get a bit smaller and less firm. Sperm production usually decreases, though many men remain fertile into old age. The prostate almost universally enlarges, leading to BPH risk, as we discussed. Ejaculatory force and volume often decrease. And importantly, related to sexual function, the blood vessels, arteries, and veins within the erectile tissue can undergo sclerotic changes. Hardening, basically.
Like atherosclerosis elsewhere in the body.
Similar process, yes. Reduced elasticity, potentially narrowed lumens.
And that ties directly into erectile dysfunction, ED.
It's a major contributor. ED is defined as the persistent inability to get or keep an erection sufficient for satisfactory sexual performance.
And the mechanism, especially with aging or chronic diseases like diabetes or hypertension,
is often vascular insufficiency. If those sclerotic changes impair blood flow into the pudendal arteries and then into the corpora cavernosa,
you can't get enough blood in or trap it effectively to achieve full rigidity.
Exactly. The erectile tissue just can't distend properly. It's fundamentally a blood flow problem in many, many cases.
Okay, let's try and wrap this up. Key takeaways for the listener.
I'd say there are three main pillars to remember. First, the absolute necessity of precise temperature control for making viable sperm. That 2-3 degree difference is critical.
Right, the scrotum's location and cooling mechanisms.
Second, the whole system runs on that finely tuned hypothalamic pituitary gonadal axis, GnRH, LH, FSH, testosterone inhibin, and that crucial negative feedback loop.
Keeping hormone levels balanced.
And third, the dynamic interplay of the autonomic nervous system for sexual function. Parasympathetic for erection via nitric oxide, and sympathetic for emission and ejaculation.
Point and shoot.
Point and shoot. Understanding those three pillars helps make sense of both normal function and the common dysfunctions we see.
Okay, so we've traced this intricate system from its embryonic origins through its complex hormonal and neural controls to the changes that occur with aging and disease. We saw how development hinges on that timely hormonal cascade of T and DHT.
Right.
So, connecting it all, here's a final thought to leave you with. We heard that low testosterone and conditions like ED are increasingly recognized as early predictors, almost like canaries in the coal mine, for serious cardiovascular disease.
Strong link, yes.
So the provocative question is, could optimizing male hormonal health, or at least monitoring it more closely, become a more mainstream strategy? Not just for treating symptoms like low libido or ED, but as a potential frontline defense or screening tool against systemic illnesses like heart disease. Should hormonal health be part of preventative cardiology?
That's a really interesting question, linking endocrinology and cardiology more tightly. Definitely something to think about in terms of future preventative health strategies.
Food for thought.
Absolutely. Well, thank you for joining us on this deep dive. We hope breaking down the male genitourinary system this way helps you connect these intricate mechanisms to their clinical significance.